Yeah, so today I want to talk on food testing. This is the part which usually come in paper one, biology, paper two, and also paper three under practical. So please pay attention and understand the procedures and also the food sample concept. So we talk about the food sample. We're talking about the testing the presence of a given sample. We need to know that there are seven classes of nutrients. So the first, so for example, we have the name that I use myself to memorize is Caliproviro. So this is the way that I use myself to identify the seven types of nutrients. So the Ca stands for carbohydrates, lipids, proteins, water, mineral salt, vitamins, and roughage. So these are the seven classes of nutrients. So I've got one protein, one carbohydrates, two lipids, proteins, water, mineral salt, vitamins, roughage. How many are they? 1, 2, 3, 4, 5, 6, 7. So these are seven nutrients, food nutrients. So now, we need to carry out a test to know which food sample is present in the component given. So, what we call the food tests. So the first one is testing for starch. Testing for starch. Testing for starch. Yeah. So testing for starch, we need to know if the food sample given contains starch or has no starch. So there's some procedure that we're supposed to follow. The procedures. So before we go to the procedures, first we need to know and understand the type of food that contains starch. So talk about starch. So starch mostly they come from plants. Example like maize, which is meal, rice, potatoes, etc. So these the most come from plants. So example of starch we have meal, which is a shima, meal, we have potatoes, we have sweet potatoes, etc. So these are usually come from plants. Then now we need to understand that the chemical that is used to test for starch is either iodine solution, iodine solution. So if they ask you the name of the chemical which is used to test for starch, the answer is iodine solution. This iodine solution, it is brown in color. So it's brown in color. So I said the chemical that is used to test for starch is iodine solution, and it is brown in color. Okay, so the solution is brown in color. Then when testing for starch, always use drops. You use drops. So for iodine solution, we use drops. Don't forget, iodine solution we use drops. Then the color for iodine solution is brown. Example of food that contains starch. These are carbohydrates. So any carbohydrates, for example, like in meal, shima, potatoes, etc. All those they contain carbohydrates, and they will contain starch as well. So I'll go to the testing for starch now, the procedure. So here we need to understand that sometimes they can give you the sample in liquid form, or sometimes they can give you a sample maybe in solid form or not in liquid form. So you can still carry out the test for starch. So here I'll start with a solution form. Or for example, when the solution, a liquid form is given and they ask you to test for starch. So I'll start with the method here. Method. So on top of the method, this is a procedure how you're supposed to carry out the test for starch. So number one, the first step we say you put Okay, so from here you put 2 centimeter cubic of sample solution. For example, like if the sample solution P or sample solution P is given, maybe you can name it P or K or whatever. So sample solution P in a clean and dry test tube. So it means you put the sample solution given in a clean and dry test tube. Then from there, after putting it in a test tube, the solution, then you now add 2 drops of iodine solution. So here, if you want, you can say add 2 or 3 drops. You can say add 2 or 3 drops, but be specific. If you want, I can put 3 drops here. If you want, I can put 4 drops here, but 2 or 3 better. Okay? So method, you put 2 centimeter cubic of sample solution P in a clean and dry test tube. Then from there, you add 2 drops of iodine solution. As I said, iodine, we use drops. Then from there now, you observe, which is observation, you observe what will happen. So if the solution turns black. So here, mostly the color that will change if the starch is present is blue-black. So blue-black, but it's possible you can only see one color, so there's no problem. You can write the final color that you have observed with your eyes. So it can be blue or black. Blue or black. Say blue or black. Or blue-black, Blue or black, yeah. So it's possible that you can write the final color that you have observed. So if starch is present, the color of the solution will turn blue-black. And if starch is absent, you say the solution remains brown because the color of iodine solution is brown. So if the starch is absent, you say the solution remains brown. Okay. Then conclusion. Now we say conclusion here. Conclusion. So since here the starch is present, so you say starch present, which means the starch is available. Starch is present in the food sample given. So this is the method here that you can use to test for starch. So conclusion: if the solution here turns blue-black, conclusion will be starch present, and if the solution remains brown, which means the starch is absent, and you say starch is absent here on the conclusion part. Okay. So now, what if the solution, if the particle, example like the solid form is given, for example, like they give you, maybe if they give you millimole here, and ask you to even banana or orange, and ask you to test for starch. So what you can do, since this is millimole, this solid particles, you can just say get small sample of solution P, small sample, just get a small sample and place it on the white towel. So get a white towel. So get a small sample of this substance. You place it on the white towel. Then you just add 2 drops of iodine solution. So if the color again turns blue-black, which means starch is present, and if the color remains brown, which means the starch is absent. So don't forget, in case if the sample given is in powder form, you just get a small sample of this, put it on the white towel. So on the white towel, then from there you add 2 drops of iodine solution, and if the color changes blue-black, starch is present. If the color remains brown, starch is absent. Okay. So the next part is how to test a leaf for starch. Testing a leaf for starch. Starch. So for example, how can you carry out a test for leaf for starch? So testing a leaf for starch. Okay, so you get a leaf from a plant, and I know that when you get a leaf from plant, the leaf or the metabolic reaction inside the leaf will not stop immediately. So after getting the leaf from the plant, the first stage that you boil the leaf in warm water. So you boil the leaf in warm water here. You boil the leaf in warm water in order to stop all cell activities or to keep protoplasm. Because after getting the leaf from the plant, the leaf or the chemical, the metabolic reaction inside the leaf will not stop working immediately. So in order to kill all cell activities in the leaf, what do? You boil the leaf in warm water. Then after removing the leaf in warm water, you boil it in methylated spirit or alcohol. You can use the methylated spirit, which is this one, right? This one here, or you can use alcohol. You can also use alcohol. Then now you don't boil direct. You use water bath here, water bath. Then you put your leaf in a test tube there. Then you close the mouth of the test tube with cotton wool because we all know that alcohol is inflammable. So to prevent this alcohol, which is inflammable, to cause accidents, we close the mouth of the test tube with cotton wool. Okay. Then immediately after removing the leaf from the test tube, I mean from the alcohol, the leaf becomes brittle. Brittle means if the leaf is alcohol says it will be hard and it will be impermeable to iodine solution. So the best way after removing the leaf from alcohol, you also you put or dip it in warm water in order to soften it. So after removing the leaf from alcohol, you dip it in warm water in order to soften it. Then now you get the leaf, you place it on the white towel. Then you add 2 drops of iodine solution. Then if the leaf turns blue-black, the starch is present. And if the leaf remains brown after adding iodine solution, then the starch is absent. Then again, what's the purpose of dipping the leaf in alcohol here? So the purpose of dipping the leaf in alcohol is to remove chlorophyll, that green part or green pigment from the plant. And that green pigment can disturb or interfere our experiment or disturb our experiment. So we boil the leaf in alcohol in order to remove chlorophyll. So these are the 4 stages: number 1, number 2, number 3, and number 4 stages when testing a leaf for starch. So I'll go to testing for protein now. Protein testing. I'll go for protein. Okay, so before I go to the testing for protein, first one we need to understand the type of food that contain protein. So protein, if I to have examples, we have eggs, we have beef, also have eggs, beef, fish, etc. All those they contain proteins, and proteins are very important in our body. So sometimes they can ask you the importance of protein in the body, the importance of carbohydrates. So pay attention. The importance of protein, the importance of carbohydrates in the body. Then at the same time, what disease is caused when someone is lacking protein or carbohydrates in the body. So I want you to research on this part. The type of disease which is caused if protein or carbohydrates is lacking in the diet. Apart from that, what are the importance of protein and carbohydrates in the body? Okay, so when testing for protein, we use what we call the chemical that we use is the burette. So burette, it's the blue in color. It's the blue in color. Okay. So this one we don't use drops, but we use centimeter cubic to measure it. And this one always requires requires the sample to be in solution form. So if the sample is not given in solution form, which means we need to extract the sample given and convert it into solution form. How? So by getting the small or getting the sample given and grind it by grinding it. After grinding it, you mix it with the distilled water. Then now you filter the water, and after filtering, you get the liquid. Now you test it for protein. So I'm saying that this one requires the sample to be in solution form. So if the sample is given in solid form or in powder form, make sure that you grind the sample given, mix it with distilled water. After mix with distilled water, then now you filter the water and get the same liquid and test it for protein. So now what are the procedures? So procedure now, since after extracting the liquid, we have now a solution, the solution. So you say the method now, method here, the procedure, the method, right? The method. So the method number one, you put 2 so if you want, you can use the 3 centimeter cubic here, 2 centimeter cubic of sample solution, okay, in a clean and dry test tube. Then add, so you add equal volume. So here, if we have 3 centimeter cubic, even for burette we put equal volume, which is 3 centimeter cubic. If you have 4, even here we put 4. If you have 1, even here we put 1. So you add 2 centimeter cubic of burette reagent. Then from here now, observation, you observe what will happen. Eh? So if the solution turns purple, so solution turns purple, which means conclusion will be a protein is present. So if the solution turns purple, which means the protein is present, and if the solution remains blue, which means the protein is absent. So don't forget these colors, are very important, because even in paper they can ask you, for example, like what will happen if the protein is present after testing. So which means the solution turns what? Purple. And if it's absent, the solution will do what? Will remain what? Blue. Then for starch, if the starch is present, it will turn blue black. And if it's absent, it will turn what? It will remain what? Brown. Present, blue black for starch. Absent, it will remain what? Brown. Then for protein, if it's present, it will turn purple. And if it's absent, it will remain what? Blue. So this is the first part that I wanted to explain. The next time, I'll explain on the testing for reducing sugar and other components. Thank you very much.